super excited for this next talk. Although I'm a bit biased, I work with Jasmine, but Jasmine is like such a fantastic theorist and experimentalist, and we're going to hear about some really exciting things about complex biological systems. So please take it away.
Thanks, Maria. So thanks, guys, for putting together such a super workshop and gathering together this fantastic group of people. It's really, really exciting to be part of this lineup. Okay, so in our lab, the kind of research process generally goes like probably something that seems similar to a lot of you. We see something that's fascinating or it's unexplained in the field, or we see a weird result in some paper in the literature. And then this opens up a ton of new questions in our brain, and then we set out to figure out what's going on. And often our kind of first attempt, sometimes that's, building a model, or sometimes that's doing a series of exploratory experiments. And this always lands us somewhere crazy. And then we have to gather ourselves and enter this back and forth process between theory and experiment until we finally get to somewhere that can satisfy us. Yeah.
Nice.
So I included this video because one, I think it's pretty illustrative of this process of going back and forth between theory and experiment, and also because it's an experiment that's currently taking Alistair to some pretty fun and crazy places, I think. So the questions that we ask center around how organisms adapt their locomotive strategies to deal with environments that can be heterogeneous or that are constantly changing and throwing a bunch of dynamic cues at them. And I'm saying this as kind of a pretty purposely really broad sweep, because our research, like many of yours, is largely curiosity driven. So this means we end up working with a ton of different organismal systems, from bacteria to tardigrades to jumping spiders, as you saw earlier. And even within the same system, there are a ton of fascinating questions that span scales, span levels of organization, and these require massively different experimental techniques and theoretical techniques to tackle. And so this is actually the case with the two stories that I'm going to focus on for this talk, which are both about how E. coli manages to navigate complex environments like soil and the inside of our guts, with adaptations at two different levels. So in terms of whole cell behavior, and then zooming in to talk about the actual molecular machinery that drives the locomotive process. The brief intro. So E. coli swims using something called run and tumble motility. And in unconfined media, it looks like this. So bacteria have a bunch of flagellar filaments that form a bundle when the motor is rotating counterclockwise, and that makes them go in a straight direction for a period of time. And then when the motor switches direction, they go, their bundle splays apart and they enter this tumble, after which they choose a random new direction and go enter a new run. And if we want to quantify this, by the way, if anybody has any questions, just yell it out at any point. I'm not married to this being a linear talk. Okay. So if we want to quantify this at the ensemble level, we can look at the mean squared displacement, and run and tumble shows ballistic behavior at short time scales, which corresponds to the length of a single run. The average length of a run in unconfined environments is about 2 seconds. And then it shows transition to diffusive behavior at longer time scales. Now, when bacteria, E. coli, are faced with chemical cues from their environment, so food, a gradient of food, a gradient of toxins, they can lengthen or shorten the length of their runs, increase their tumble frequency to go in a direction they like or go away from a direction that they don't like. But in contrast, what they do when they're faced with any kind of mechanical stimuli in cluttered or complex environments is massively, massively understudied. And this is true even though most bacteria, including E. coli, spend very, very little of their actual lives in unconfined liquid media, like in the lab video that I'm showing here. And so there have been a few theoretical studies on what might happen in cluttered environments, and maybe an obvious kind of first pass guess is what happens is similar to chemotaxis, right? So their run lengths get shortened because they keep hitting obstacles that block them before they can complete their run. And quantitatively, what we would expect this to look like is you'd see a lower crossover time between ballistic and diffusive periods because your run length is getting short. But when there were finally kind of a set of clear experiments done in this regime, things look really, really, really different. So this is some experiments from Sujit Datta's lab done at Princeton, now at Caltech. And this E. coli is moving in really tightly packed hydrogel. And it's doing something that looks so different from run and tumble that they just like gave it a new name. And they called it hop and trap. And quantitatively, as you might expect, the dynamics also look really dramatically different from what we'd expect. So there's a shortened or close to non-existent ballistic region, which we might expect from a shortened run. But we also see these transient subdiffusive dynamics at intermediate lags. And so what we set out to ask is, are these two actually different locomotive modes? So are the rules of motion that individual bacteria are using, is the motility program fundamentally different in these two conditions? Or are run and tumble bacteria just exhibiting these like radically different observed motility patterns, doing the same exact thing, but just in radically different environments? And then to start tackling this, we started thinking about, transitions between these environments. So what happens in the middle? After all, like E. coli in the real world are interacting with a whole host of different environments that span open fluids and super tightly packed soils or hydrogels. So to get at this, my grad student, Haibei Zhang, and a collaborator, Miles Wetherington, took the first jump here with a series of exploratory experiments. And they designed a microfluidic chip to expose bacteria to a wide range of different structured environments. And then they can characterize motility patterns across these different conditions. So here are some E. coli that are moving through one of the regions of the device with tracks laid over them. And the device also contains an unconfined region with no obstacles as a control so that we can compare properly. And then as for the structured environments, each one has pillars that are arranged to a particular level of confinement, which corresponds to the mean spacing of the pillars, and a level of disorder, which corresponds to the variance of the pillars from grid spacing. So for example, the environments can range from something with a sparse grid, like it's C equals 6 microns and D disorder is 0, to something that looks like a really tightly packed disordered arena at C equals 1.3 and D equals 3. And these are bacterial trajectories moving through each of these.
Like this Manhattan-like distance is real? Those are real data?
Yeah, these are real data. These are real bacteria. Super weird. And so after looking at trajectories across all these regions, what they found was some of the trajectories resemble classical run and tumble, and some of them resemble hop and trap, and then kind of everything in the middle. And the proportion of things that look more like hop and trap change smoothly as the arenas get more and more confined and more disordered. And so, okay, what this first pass showed us is that there's not really a sharp transition between these two dynamics. Bacterial motility patterns, at least observed ones, seem to lie on this continuum. But of course, like the observed patterns that we can image are convolutions of environmental structure and bacterial behavioral decisions that they're making through some internal chemistry. And just to kind of briefly go over what that chemistry is, again, I told you a little bit about run and tumble, and at each flagellar base, there's a motor, which I'll talk more about in the second part of the story. But when this motor rotates counterclockwise, the bacterium is executing a run, and when it switches to go clockwise, the bacterium initiates a tumble state. And in unconfined environments, there's this clear one-to-one mapping because there's nothing in the environment that's preventing the bacteria from expressing like its true internal desire. And so everything that we see is what exactly is going on inside the bacteria.
That's a super stupid question.
Yeah, totally.
So the paradigm of like this mean square displacement versus time makes sense from the perspective of Brown, but for these sort of other systems, like first passage time distributions that all tell more intricate discrepancies between these things? Because you have the full trajectories.
Yeah.
So it could be like t to the minus 3 halves or not. Like,
yeah, no, that could actually give us more information, but this was kind of a first pass analysis of like what would happen across this, but that could be like a very interesting future direction. But yeah, so you can kind of see this in this video too, that in unconfined environments, when a flagellar bundle flies apart, bacterium stops moving forward and enters this tumble. And when they're moving forward, they're in this kind of tight bundle behind them. But in confined environments, we don't know what this mapping looks like, right? So do bacteria change their behavior like they do in chemotaxis to optimize motility to these different environments? So do these observed transitions actually correspond at all to internal transitions? Or are bacteria essentially doing nothing different, and then the changes that we observe are purely driven by the environmental structure? So for instance, if flagella get trapped in this tight spot and they can't splay apart, even if the internal chemistry says, let's initiate a tumble, or if they get stuck in this kind of dead end after a tumble and they can't move forward after the next. So we can't really get at internal states like this just from imaging the cells, and finding out this mapping experimentally is really, really challenging. So this is when we kind of start turning to theory. And we computed the chord length distribution, which looks at the maximum distance that a straight moving particle starting at some random point can go before it hits something in its environment, in any given environment that we can map out. And then looking at the distribution of expected run lengths, which are taken both from run and tumble simulations and from behavior in the unconfined environments, we conditioned on this chord length distribution, and we found strikingly good agreement in, this is from one of the environments that you can see the mapping of this dotted line on top of the solid line. And so this suggested to us that the run and tumble behavioral program is robust to environmental perturbation. So that is, E. coli aren't really changing anything about their underlying behavioral program to deal with navigating all of these different environments. And they're moving through all of these structured environments without really changing anything about their internal chemistry. And then now we go to thinking about, okay, we have this really robust evolutionary motility strategy. What is the underlying machinery behind it? And so for the second part of the story, we shrink ourselves a little bit to think about bacterial swimming from the tinier perspective of the molecular machine that I've mentioned a couple of times that drives flagellar rotation, which is the bacterial flagellar motor. And I promise I'm not going to go through all of these proteins, but just know that the motor consists of 3 general parts. There's a flexible, do you see my mouse? Okay, good. There's a flexible hook that connects the flagellar filament to the basal body of the motor. And the basal body consists of a rotor. It's called as such because it rotates with respect to the cell body. And it has these series of rings that go through the entire cell wall of the bacterium. And it has these stator units with these MOT units, which each connect to the peptidoglycan layer and then are stationary with respect to the cell wall. And they generate torque at this stator-rotor interface by interacting with these Fly-G protein spokes along the C-ring and rotating it around. So when I first started to think about this motor 10 years ago, I wrapped my mind around it at first by thinking of a macroscopic analogy, and I thought that the structure really lends itself very nicely to that of a merry-go-round like this. The rotor is ring-shaped. It has all these protein spokes around it. We can be like, these kids on the playground that are pushing it around to help our friends, just like the stators do for the bacteria. And as you might expect, for a bacterium that has to navigate tons of different environments, the motor has a really wide functional range, and it spans kind of open fluid when there's very low torque on the motor because flagellar filaments are really thin and don't impart that much torque on it. And high torque when bacteria are in really tight confined spaces or in a dense collective of bacteria or in very viscous fluids. And an important thing to note is that a single motor is often kind of spanning its entire functional range in very quick succession, right? Because they're dealing with really complex biological environments that are transitioning between these regions very, very quickly. And what that means is that the flagellar motor has to be able to make these transitions very quickly, to sense and respond to mechanical changes from their environment, or deal with not needing to sense and change. And perhaps surprisingly, in relation to the kind of first half of this story, we found that the flagellar motor actually does actively respond to changes in its environment. It was observed in a previous study that in E. coli, motors that are operating at very high load have more stator units engaged to them than motors at low load. But we didn't know anything about the dynamics of these transitions. So is this something that, if you evolve E. coli in a very high load environment, they will recruit more stators and have higher torque motors? Or is there some sort of dynamic transition that happens in real time between these two states? So thinking back to kind of the playground, how would we deal with these kind of switches, right? So let's say, just our best friend wants a push. Now, this is not too challenging. We can make the motor really go fast, and we don't get that tired. Everybody has a lot of fun. And then after some time, lots of more people see how much fun this is. They all want to get on the motor. They all want to get on the merry-go-round. And now your arms are starting to get really tired. You slow down, and maybe you call some friends over to help you push. And so to figure out if this is kind of what was happening, this time we took our first jump with building a mathematical model of stator engagement and disengagement of the rotor, characterized by an on rate K on and a disengagement rate K off. And what we aim to ask is the arrival rate or the leaving rate or both sensitive to the environment? So when do you decide to call your friends over for help? How do they decide when to come over? How do they decide how long to stay? And then once we had the model in place, we wanted to do some testing of it, some validation. And to do these tests rigorously, we designed and performed some experiments in the lab. And one really cool way to get at motor dynamics and fit this kind of torque speed curve is to do something called a bead assay. And you stick a bacterium down to a slide, you shear off its flagellar filament, and you stick a bead onto the stump. So you can get the speed out of the bead from a video or using an optical feedback trap. And then because these beads are really small, you're operating a very low Reynolds number, and then you can easily calculate out the drag on it, which is equivalent to the torque that the motor has to apply to rotate a bead of radius r at some speed omega. And so this gives us a picture of what the motor is doing at a particular load on this curve, noted by this red bar. And the position of that is determined by what size bead we use. So if we use a smaller bead, we move towards the lower load, higher speed region. We use a bigger bead, we get over to closer to stalling the motor. Oh, yes, hi, sir.
Question: how did you attach the bead to the flagella?
Yeah, so there's multiple ways. You can coat the bead in something sticky like polylysine, or we have a bunch of bacteria now that are genetically modified to express biotin on the end of their hooks, and then we have streptavidin beads that we can attach on. So each of these gives us like a static picture of what's going on at each one of the points of this curve. But we still don't really have an idea of the dynamics of what's going on back and forth, because biotin streptavidin links in particular are really, really strong. And once a bead is on there, you're not going to take it off and attach a different bead on. So we had a little think about this. And what we thought about was, okay, instead of using plain polystyrene beads, what if we use superparamagnetic beads? Then we can manipulate and perturb what the bacteria are feeling by using external magnetic fields and not changing the size of the bead at all. So in the first pass, we used tube-mounted permanent magnets that exert some torque on the bead. And for this experiment, we lowered the magnets to a height so that we get to stall torque. And then we leave it there for 5 minutes. And then we remove it and we let the motor relax back. And you can see here that once we've removed the torque, there's a big jump in stator number. And when we remove that torque, the motor undergoes subunit exchange again and relaxes back down to the original stator number for this particular beat size. And from this relaxation process, we can get out characteristic time, steady state stator number, and then we can plug that back in to that ODE model that I showed earlier to get out an idea of what engagement and disengagement rates are. And then we can repeat this process for different bead sides, which basically what this does is allows us to get an idea of how these rates look as we go across this curve, right? So the magnets pull us over to stall torque, and then once the beads are released, the relaxation processes take us to various points along the curve. Okay, so I have a little time left, but I'm just gonna skip over a little bit. But we can't really get to the low load using permanent magnets just because of the size of commercial beads that are available. So what we ended up doing was, instead of just using permanent magnets, we used rotating electromagnetic fields that could take us back and forth across the entire thing. Yeah. And
5 minutes because we're a little late. So like 5 more minutes.
Okay, cool.
Yeah.
No, I have an end which I want to like have open questions that like I would love for people to like talk about. So I just want to. Okay, so we have magnets. Boom, you can get this in real time. This is a single cell instead of an ensemble. So that's fun. And that allows us to kind of get back and forth across this entire curve. And putting all of that together, what we found was that the on rate, the engagement rate shown in blue, wasn't really dependent on external conditions, but the off rate, which is shown here in red, decreased as external load increased. And so stator units are coming by to check on their friends all the time, regularly, despite how tough the job actually is. But if they see that they're needed, then they stay for a little while. And this kind of bond, which we heard about this morning, which grows stronger with larger external force, is called a catch bond. And it's something that's, really widespread molecular strategy. And as we heard about already, and a lot of systems from bacteria to eukaryotes use this to sense and respond to mechanical stress in their environment. And because I really like a macroscopic analogy, a good way to think about this is also one of these finger traps that you put your finger in. And if you struggle and you pull, it gets tighter and you can't escape. And the only way to actually be able to get out is to just relax and let the bond go slack. And this is a strategy it seems like the flagellar stator is also using, but learned it over evolutionary time. All right, so now we've kind of looked at the same question, right? How do E. coli adapt to move through complex environments? Do they adapt to move through complex environments? From two different perspectives, and we kind of got two different answers. And one thing that or at least I have been really excited about, is how do the adaptive dynamics of these two different length scales interact with each other? So for instance, let me go back to this torque speed curve. It's been suggested experimentally that the switching rate of the motor, which corresponds to the tumble frequency, is dependent on motor torque. And mechanosensitive motors, or at least if in E. coli you remove mechanosensitivity, you lose this torque plateau. So how kind of motor mechanosensitivity is related to kind of maintaining constant tumble frequency, unclear. We don't really know how those two dynamics relate to each other, but it seems just from a very kind of first pass analysis, like first pass thought of it and like a back of the envelope calculation, that it might be important. Adaptation at the motor level might be important for maintaining tumble frequency across different environments. And also then zooming out kind of beyond E. coli and looking more broadly across bacteria, and the flagellar motor and flagellar swimming is a prevalent way of getting around across different species. And we can ask these kind of very similar questions, like what are the trade-offs of being adaptive at either level or both? What are the environmental features that favor the evolution and maintenance of adaptive motility strategies? And how do adaptive strategies on shorter timescales? This is kind of a very like broad pie in the sky question that I really want to ask, like across systems, not just in bacteria. How do adaptive strategies on short timescales affect structural evolution on longer timescales. And you can think about this in terms of, you know, if I'm really, really good at adapting to changes in my environment, does that remove selective pressure on me to evolve and get better structurally? Or does it mean that now I explore more niches and I actually diversify more? And I don't know that there is a singular answer to this, like across the tree of life, but it's something that I've always been very, very curious about. And I think bacteria and the flagellar system in particular are a really nice system to test this out experimentally and possibly theoretically on. And just as a kind of example of this, like there's really just such a broad diversity of flagellar motors. So I mean, they're not as weird as Ben's protists, but I think like, you know, if you really, really look across the bacterial tree, there is a flagellar motor for everybody. And there's like versions of the mechanosensitivity that act like slip bonds. There's versions of mechanosensitivity that are really, really difficult to activate. So they're ones that kind of hold on to all their stators all the time. And how these have evolved in different environments, we know so little about, because we've been really limited by structural studies until very recently. We got our first structure of the flagellar stator at molecular resolution like 3 years ago. And then also at this level, run and tumble is the most well-studied form of bacterial flagellar motility, but it's not the only one. There's a whole host of really interesting ways to move, one of which is called push-pull-wrap that Vibrio fishery uses. And this is you can see push and pull here, and they also have this very interesting mode in which the flagellar filaments wrap around the bacterial cell body. And this mode is really, this kind of mode of motility is really helpful for fishery in particular, because they are a marine bacterium that also is a symbiont of the squid, of the bobtail squid. So it lives part of its lifespan within squid tissues and part of its lifespan swimming in the open ocean. And accordingly, we see a clear adaptation, or this is from a paper in 2024, which is not showing there, but by a Taiwanese group, which is a fantastic paper, first characterizing how this mode of motility changes to increase the time spent in wrap when viscosity increases, which really helps them channel through squid tissues. All right. Okay, with that, I will stop. This is all work done by really fantastic lab members and collaborators. So thanks to all of them. And then I'd be super happy to talk about any of these at any time.
The first part, MSD. So you know you said confinement. I think it solves most of the topology of confinement most matter, because the pillar and the pusher will just go around the pillar.
Yeah.
So then if you look at the MSD, it looks like, even though it's for the bacteria, it's a running motion.
Yeah.
In the MSD analysis can be sort of the stack, the stall in the one location, right?
Yeah, absolutely.
So have you tried to look into something like?
So that kind of like motion happens even because it's a quasi 2D environment. So we get that kind of circular motion even in the unconfined region when we have our device. We remove most of those circular trajectories to kind of to not have that confound it. We haven't explicitly looked at what happens when we add those back in, but for that reason, we take out the trajectories where we have, or the portions of trajectories where we have lots of circular.
Confinement. Have you tried to change whether it's pill-like a structure or like the topology of the structure doesn't matter?
So we haven't changed the topology of the structure, but there was a recent study. I can send you the paper. I forget what group it was from, that showed that the geometry of the pillars don't matter. They do, bacteria, if kind of given the surface, they'll follow it, but even if you have oddly shaped pillars.
Are there any types of bacteria that have multiple types of flagella?
Yeah. So Pseudomonas has the same type of like the flagellar filament is the same. So the flagellins are the same in the flagella. but they have two different types of stator systems, one for high torque stator, one for high torque situations, and one for low torque situation. And the dynamics between switching between those systems is on a much longer time scale than remodeling. So, but Pseudomonas is like particularly finicky to work with in terms of the bead assay. So we haven't like successfully gotten a good bead assay working on it yet. But understanding the kind of like interaction between mechanosensitive remodeling of the kind of seconds time scale and then this remodeling between the two stator systems is something that's like in the back of my head. Yeah, it's an awesome system.
I'm confused about this is lovely talk.
Yeah.
Thank you so much. I'm confused about the sort of bioenergetics of this motion. So if I understand correctly, from a perspective of like dragging a thin small object, that like the first principle bound on how much energy it would require to move it Stokes drag was not that much.
Yeah.
But then if you ask like how much bacteria actually spend in this type of motion, like they spend like 1 ATP per, or like one.
So they don't spend any ATP on it. So the rotation is purely ion. It's driven by ion passages.
They to maintain the ion gradient.
To maintain that.
Yes.
Yeah.
So I guess, is this efficient? Is this not efficient? Do you have any like way of how to like in the cartography of the adaptive or like the sort of adaptive motions that you're describing? Is there any sort of real physics knowledge about the sort of cost of each of these modes, or is this still?
So it's hard because the ion gradient is maintained for lots of different purposes, right? So the energy of maintaining the ion gradient isn't the energy of powering the motion, but if you calculate kind of the energy that's used per ion passage, it's a remarkably efficient process.
Well, for rotation.
For rotation,
but not for
but also for swimming. It's a efficient process to move. They don't use. I'm trying to think about there was a paper that kind of did a back of the envelope assessment of what percentage of their energy budget is locomotion. And it was surprisingly low. It was like under 10% or something. But the challenge is that making flagella and maintaining flagellar filaments is super expensive. And they have to do it often, right? Because when they divide, they just split their flagella. So then they're constantly building flagella. And that bit is really expensive. So.
Thank you.
One last question.
Okay. So for these flagella to bundle and unbundle.
Yeah.
So when they bundle, for example, there are a bunch of motors working together.
Yeah.
But for each one of these motors, do they feel more torque or more resistive force on the other end, so that they have to recruit more protein to move it, or do they not change the number of proteins they need?
Do you mean that the more flagella are in a bundle, does each motor feel something from the hydrodynamics of the size of the bundle?
Right, yeah.
Probably. And so this is also something that we've always been really curious about, is how the switching rate changes as, sorry, you can disconnect. How the switching rate changes as a function of flagella number. It's a completely unknown and super fascinating problem, but I think the answer is yes.
I see. Thank you.
I guess during this transition, if I guess we can probably do one more question as our transition, but I don't know. Or
I think
otherwise,
Jasmine, one more time.
Yeah.